Welcome students. In today's session, we are going to start with the second unit of our syllabus, that is bioelectric potentials or physiological signals. Okay, so in this unit, we are going to cover the topics action potential and impulse propagation, origin of bioelectric signals, electrode theory, type of electrodes and selection criteria, recording electrodes and skin impedances, electrical conductivity, and microelectrodes. So this particular unit is going to deal with the electric signals that are being produced by our body, and we will see what are the types of these signals that are produced, why they are produced, and what type of electrodes, what different types of electrodes are there that we can use for measuring the bioelectric potentials, right? So first of all, we need to understand what is bioelectric potential. Now, bio plus electric means is an electrical signal which is related to some biological phenomena, and potential means voltage. So over here, we are going to talk about the electric voltage that is being produced in human body due to several various phenomena. Okay, so the first and foremost thing that we are going to talk about is the potential that gets developed at the cellular level. That means at the cell level. So there is an electrochemical activity taking place at the cellular level which actually gives rise to certain ionic voltages. Okay. So bioelectric potentials, they are actually ionic voltages that are produced as a result of electrochemical activity taking place at the cell level. Okay, these signals, they are measured using transducers that are capable of converting the ionic potential into electrical voltages. So we have at the cell level, we have a potential difference, an ionic charge, you can say accumulation of charge in such a way that a potential difference develops over there. And we need to have certain transducers, which of course we call electrodes in this regard. We have specifically designed electrodes which are capable of converting this ionic potential into the electrical voltage, which can be used in any circuit for further processing. So we need to understand from where this potential is actually coming. So we all know that the cell is actually made up of a semi-permeable membrane. You know, there are a lot of things like there's a nucleus, there's mitochondria, and a lot of other things which are there inside the cell. And all these things, they are encapsulated in a semi-permeable membrane. Okay. Now, when I say semi-permeable, it means that the inherent behavior of this membrane is to allow certain ions to pass, whereas to restrict others. We all know that within and outside the cells, it is all full of fluids, and these fluids, they contain ions like sodium, potassium, chlorine, etc. So the cell membrane is actually semi-permeable in nature. That is, it will allow certain ions to pass through, but it will not allow all of them. So some of the ions may pass easily, some may not. Okay. So, in the cell membrane, it has a tendency to permit potassium and chlorine ions very easily. Okay. But it has a tendency of restricting sodium ions. So sodium ions are positively charged, potassium ions are positively charged, but chlorine ions, they are negatively charged. Okay, so due to this, but potassium ions actually their mobility is less as compared to sodium. Okay, so basically, the if you see the figure on the screen, you can see that the concentration of sodium is actually low inside the cell and it is high outside the cell, right? So this high concentration actually results in an accumulation of a positive charge at the outer circumference of the semi-permeable membrane and a negative charge at the inner circumference of the membrane. So if you see in the figure, just near to the surface of the membrane, you can see outside it is full of positive charge, and the inner boundary is actually covered with negative charge. So this positive and negative accumulation of charges is actually giving rise to some potential difference. Okay. So this makes the outside portion of the cell more positive as compared to inside. So to balance this potential, the potassium ions that are also positive, they also try to enter the cell. Whenever you see there is a potential difference, there will always be a tendency of ions to move. So when there is a more positive accumulation of charge outside, the positive charge will obviously try to move inside to achieve some kind of an equilibrium. But since sodium ions cannot move very freely, the potassium ions, they are also positively charged, so they try to move inside the cell. However, a perfect balance is never achieved. This is because this is a very dynamic thing. So perfect balance is never achieved, and a sort of equilibrium is developed when the potential difference across the membrane is developed. It is negative on the inside and positive on the outside. So the attractive forces of this positive and negative, actually they keep them stuck to the membrane, and so a sort of an equilibrium is maintained. Right. So this particular potential, this which is developed across the membrane, is called the resting potential of the cell, and it is maintained until some kind of an external disturbance is applied which upsets the equilibrium. Okay, so we can see from this figure that due to the semi-permeable nature of the cell membrane and variable movement and restricted movement of the various ions, we see that ultimately when the equilibrium is maintained, there is a positive charge at the outer circumference of the membrane and a negative charge at the inner circumference of the cell membrane. So this particular potential difference that has developed is called a resting potential. And since we are always measuring the resting potential from inside the cell with respect to the outer fluid, it will always be negative. So the usual range of this negative potentials is from minus 60 to minus 100 millivolts. Okay. So in this state, in the resting state, when there is a resting potential developed, we say that the cell is polarized. This kind of cell is known as a polarized cell. Now, what happens when some external stimulus is applied? Whenever you apply certain external stimulus, or you supply some external energy to the cell, the behavior of the membrane changes. Because of that additional energy, it allows the sodium ions to enter inside very easily. Okay, so when the sodium ions, they start moving inside the cell, a ionic current flow starts, and because of that energy of those ions, that barrier of the membrane, it further reduces, and there is a sudden inflow of positive sodium ions inside the cell. This is called avalanche effect. Avalanche basically means sudden flow in large quantities. So this particular, if you apply a stimulus, the membrane behavior changes somewhat. It allows the sodium ions to enter, and once the ionic current starts flowing further, the ions, they get energized, and there is an avalanche effect in which the sodium ions, they rush into the cell. Okay, now when the positive, one type of positive ions, they are coming into the cell in large quantities, to balance that effect, the potassium ions, they start moving out. Okay, but the movement of potassium ions is not that fast as compared to the sodium ions. Okay, so this results in another potential, that is a higher positive potential within the cell as compared to the cell fluid, because there is a sudden flow of sodium ions which are positive, they come and accumulate inside the cell. Potassium ions, which are also positively charged, they try to escape from the cell, but their movement is not that fast as sodium. So there is a high potential, positive potential accumulated inside the cell. This potential is called action potential, and approximate value is somewhere around 20 millivolts for a specific kind of cells. Okay, so basically when the cell is in the resting state, it has some equilibrium. The outer cell portion is positive with respect to the inner one. That is called resting potential. Resting potential is always negative because we are measuring the cell potential from inside the cell as compared to the surrounding fluid. So resting potential is always negative because there's a negative accumulation in the inner circumference. But when we apply some stimulus to the cell, the membrane, the behavior changes. The sodium ions, they get energized, and there is an avalanche effect, and suddenly the sodium ions, they rush into the cell body. Okay, and that creates a positive potential inside the cell, which is called an action potential, which ranges for about 20 millivolts. In this state, this particular cell is called a depolarized cell. Okay, so cell that is excited and it has certain action potential is called depolarized cell. And the process of changing from resting potential to action potential is called depolarization. From the figure, you can see that there is a movement of ions, potassium ions going outside, sodium ions coming inside. Okay, and the accumulation of charges actually changes. So once the flow of sodium ions, it slows down. Now, when you have given a sudden stimulus, all this activity takes place, acting potential develops. Okay, once the stimulus is removed, gradually this flow or this avalanche effect or this flow of sodium ions, it slows down and a new equilibrium is achieved. The ionic current, it lowers, and gradually the membrane, starts coming back to its original condition. So the behavior of that semi-permeable membrane gradually goes back to the same old pattern, which was not allowing the sodium ions to enter into the cell. So again, gradually, the sodium ions, the flow slows down, and there is a mechanism called sodium pump, which takes place inside the cell, and it pushes all the sodium ions quickly out of the cell, and again the resting potential is achieved. Okay, this whole process is called repolarization of the cell, right? So we have the cell at rest, it has acting resting potential. When we give some stimulus to the cell, the sodium ions, they gain energy, and there is a positive charge accumulation inside, which is called acting potential. And gradually, once the stimulus is removed, the sodium ion flow, it slows down, membrane comes back to its original condition, and all the sodium ions, they are pushed out of the cell by the action called sodium pump, and again the original resting potential is achieved. This process is called repolarization. Okay, now, regardless of any method or intensity of the stimulus that is used, the action potential that is achieved by a particular type of cell remains the same. So, the development of action potential, the value of action potential can actually be useful in identifying what kind of cell we are considering. Okay. Now, once the action potential is generated, when we had given a stimulus, action potential is generated, and that time when all the sodium going inside and all that movement is taking place, there is a brief period of time during which the cell cannot actually respond to any other stimulus. So within that small period of time, if you try to give another stimulus to the cell, it will not respond to that stimulus. So that time period up to which it is not going to respond to the next stimulus is called absolute refractory period. Okay, and just after following this period, there is again a small period in which the cell, it starts responding to new stimulus, but a very high intensity of stimulus is required. That means the response is slow. This particular time is called relative refractory period, in which the cell can actually respond to the stimulus, but it needs a very high intensity of stimulus, okay, for the cell to actually respond. So graphically, we can see that when a stimulus was given, depolarization takes place. Suddenly, a high potential, it goes into the positive. Action potential is developed. Once the action potential is developed, again gradually it comes down. Repolarization takes place. It comes to the resting potential. Then there is, after that, once this repolarization takes place, there is a small refractory period in which it cannot actually respond to any other stimulus, and then finally it again comes back to its original resting potential. So this whole activity that is taking place at the cellular level, is this is actually giving rise to the bioelectric signals from human body, and in various different types and in various forms we can tap this bioelectric signal with the help of suitable transducers, and we can get very valuable information about human body processes from these bioelectric signals. So this is the basic concept of how a bioelectric signal or a bioelectric potential is developed in human body.